Hello everyone, welcome to today's class. My name is Rituparna Palit and I am here to make you study anti-hyperlipidemics. This is from the subject Medicinal Chemistry 2nd with the code BP501T. Let's know something what we have already studied about this anti-hyperlipidemics in our last lecture. We have studied a little bit of it, like what are the drugs, what are anti-hyperlipidemic drugs, what is the meaning of lipidemia. So, what we have studied is lipidemia is a condition in which there is an increased amount of bad cholesterol in our body. That is, we have studied already that there are three types of cholesterol what we have seen, that is HDL, LDL and VLDL. What we already studied was HDL, which is also called as high density lipid, is a good cholesterol. LDL is a low density lipid, is a bad cholesterol, and VLDL is very low density lipid, that is also a bad cholesterol. So, in lipidemia what happens in our body, that the amount of bad cholesterol, the concentration of bad cholesterol is increased so much that the good cholesterol is lower or is almost you can say that good cholesterol is not able to perform its functions properly because of the accumulation of too much of bad cholesterol. So, anti-hyperlipidemic drugs or also called as hypolipidemic drugs, these are the agents which are used to decrease LDL and VLDL in our body, so that the quantity of HDL, the amount of HDL may be increased to an appropriate level, so that our body can function properly. And we also studied about the causes. What were the causes? The sedentary lifestyle, smoking, the habit of smoking, the habit of alcoholism. It is also hereditary. So, there are certain causes which causes LDL and VLDL to increases in our body so much that it hampers, it starts getting deposited in the various parts of our body, that is the vital organs are getting affected very badly with the deposition, with the accumulation of these bad cholesterols. So, these hypolipidemic drugs or anti-hyperlipidemic drugs helps us to get rid of the bad cholesterol and maintain a good functional of good functions of the organs. So, and also we studied about the mechanism of action of all the classes which we have already seen. So, the classification was we have already seen in the previous class, the statins, the secutrins and the fibrates. These are the main classes of anti-hyperlipidemics, and there is one more class, that is miscellaneous class, in which nicotinic acid, probuanol, ezetimibe and gugulipid are in this class, miscellaneous class. So, the basic three classes which are statins, secutrins and fibrates, in which the drugs which are mentioned, like in the statins we have to study about lovastatin, the thorough study of lovastatin, that means its synthesis and its SAR. In the secutrins we have to study for cholestyramine and cholestyramine, and in fibrates we have already seen the synthesis and SAR of clofibrate. That is why they are mentioned in the bold fonts. Let us continue with today's class. So, in the previous class we have already studied about the fibrates. In this class we are going to study about the statins and the secutrins. So, in statins the first drug which we need to study about is lovastatin. I am again repeating myself that we have already studied about the mechanism of action of statins, and again I would like to explain the mechanism that the glucose, that is the glucose metabolism in our body which is happening, is when the pathway, the glycolysis pathway happens. What happens? The last product of glycolysis pathway is pyruvate formation. Okay. So, when glucose that is in the glycolysis cycle completes, when the cycle completes, it converts into pyruvate in liver, and then it converts into HMG coenzyme reductase with the help of acetyl coenzyme A. Mevalonic acid, and then the synthesis of cholesterol happens. Okay. So, this cholesterol, as we have discussed about it, the cholesterol, there are types of cholesterol, good and bad cholesterol. So, we have to remove out the bad cholesterol from our body and increase the amount of good cholesterol. So, this is the full mechanism of action of statins, where what happens, the HMG coenzyme reductase mevalonic acid, this step is inhibited by statins, and ultimately there is no biosynthesis or very minimal biosynthesis of cholesterol happens. So, ultimately what statins are doing, that means the pathway which is happening from glycolysis, that is glucose to pyruvate and then pyruvate to mevalonic acid, this step is getting inhibited with the help of statins, and ultimately there is no synthesis of cholesterol. So, the drug, the first drug in this statins is lovastatin. Lovastatin and mavastatin are two different drugs in this class only, in statins class only. They are also called as new generation drugs, which were introduced into medicine for the treatment of hyperlipocholesterolemia. This is the disease for which lovastatin and mavastatins are used to treat this disease, and they are unique compounds. Why they are unique compounds? Because they are getting isolated from a plant source, from a mushroom, that is Aspergillus terrace. They are taken out. Previous to that, prior, the medicines were made with synthetic sources or from, yes, the plant sources as well, but these are special because they are taking out from type of mushrooms, that is Aspergillus terrace. The mavastatin is a chemically analog, that means there are some modifications done in the structure of lovastatin, as you can see in the structure as well, how mavastatin and lovastatin are different. The methyl group in lovastatin is absent in mavastatin. This is the only difference in the structure of lovastatin and mavastatin. So, mavastatin do not have the methyl group in it, whereas the lovastatin has the methyl group. This is the only difference, and these two agents are potent agents for treating hyperlipidemia in the class statins. So, the difference, the mavastatin is the chemical analogous, different only in the absence of methyl group at the carbon third of the naphthyl ring system. So, this is the naphthyl ring system, in which the third position the methyl group is absent in mevastatin and ultimately gives a potent compound. You can also say this is the SAR of lovastatin and meva. You can add this point in mevastatin SAR as well. So, the SAR of lovastatin and mevastatin, this is the general ring system which is responsible for the modifications and the potent activity of lovastatins and mevastatins, the general ring system, which start from first the carboxylic group will considered in the first, second, third, fourth, fifth, sixth, seventh. So, like this we are going to number the ring system and we will check what are the SAR, what is the what with the differences, what are the intermediates which are going to be made with this, what are the derivatives which are going to be made with this changes on these particular positions, and we will have potent compounds. So, the first point which says lovastatin and mevastatin, which serves as lead compounds in the development of additional statins. HMGRIs means the inhibitors of HMG reductase enzyme. Okay. So, lovastatin and mevastatin serves as lead compounds in the development of additional HMGRIs. The second thing which says all the HMGRIs, they can be chemically classified as 7 substituted 3,5-dihydroxyheptanoic acid. If you see the previous slide, the ring system, what we are talking about is the ring, is this ring system in the lovastatin, in the mevastatin, is we are talking about this ring system, because first, second, third, fourth, fifth, sixth and seventh. The seventh carbon ultimately is connected with the naphthyl group, the naphthalene ring system, naphthalene system, and ultimately gives the structure of lovastatin and mevastatin. So, the differences, the changes in this ring system can create a lots of can give a lots of potent compounds. Let us get back to the slide. So, the statins can be chemically classified as 7 substituted 3,5-dihydroxyheptanoic acid, which are subclassified based on their lower ring. If we can do modification in the lactone ring system, the bicyclic rings and the ethylene bridge can lead to the synthesis of other statins as well, like pravastatins, fluvastatins, atorvastatins, like that. There are different types of statins, and if we do the modifications in the bicyclic ring system, or you can say and the side chain of the ester, lovastatin produces simvastatin and pravastatin. That means by doing certain changes in this ring system only, not only the ring system, at the same time the bicyclic ring, that is the naphthalene and the ethylene bridge in between the lactone ring and the bicyclic ring, if we do certain changes, if we do certain modifications in these ring systems, in the bicyclic ring, in the lactone ring and the ethylene bridge, there can be production of pravastatin, fluvastatins, atorvastatin and rosuvastatins. Whereas, in the bicyclic ring and the side chain, which is this one, and this, if we can do certain changes in these two places, we can also have certain types of statins, that is simvastatin and pravastatin. So, these are the derived modifications of lovastatin and mevastatin. Pravastatin, a ring opened dihydroxy acid with 6 alpha hydroxy group, is much more hydrophilic than either lovastatin or simvastatin. If you see the structure of pravastatin, a ring open dihydroxy acid, you can consult in books for the structure of pravastatin. Pravastatin is a ring open dihydroxy acid with 6 alpha hydroxy group is much more hydrophilic than either lovastatin and simvastatin. It is a good sign. That means how much the drug is hydrophilic, that means water soluble or tendency to get dissolved in water. So, it is a good sign. So, we can give the oral medications to the patient for the in this classification, in this statins group. And the last is the if we do the replacement of bicyclic ring with substituted aromatic ring system, which lead to the development of fluvastatin, atorvastatin and rosuvastatin. It is simple if we do the modifications in that 3, which is already written in the previous slide, that if we do the modifications in lactone ring, bicyclic ring and ethylene bridge, there can be so many more newer derivatives of lovastatins and mevastatin in this classification. This is the synthesis of cholestyramine from, if we see the classification, cholestyramine comes in the class of secutrins, that is bile acid binding resin, also called as bar resin. We have already discussed the mechanism of action of for this in the previous class. How it works, like the cholesterol from liver is converted into bile and move into the intestines, and again it moves to the liver for its circulation, and this drugs which reduces the movement of bile in liver and intestine and elimination of this cholesterol through the feces, like that this bar resin or secutrant works. Okay. So, we have already discussed the mechanism of action in the previous lecture. Let us continue with the synthesis of cholestyramine and cholesterol. Cholestyramine, when styrene and 1,4-divinyl benzene, when these two gets reacted, we see a structure which is a kind of you can say a polymer, because there are so much branching and there are so much branching, and it is a big structure by synthesis of styrene and 1,4-divinyl benzene, 1,4-divinyl benzene. There is a formation of a polymer structure which ultimately gets reacted with CH2O in acidic condition and gives a bigger structure. Then it is reacting with CH3 whole thrice N, and finally we got our cholestyramine. So, if you see the structure of cholestyramine and the first two, the starting material, that is our styrene and 1,4-divinyl benzene, you can see it is the polymerized form of the conjugation of these two starting materials which are used for synthesis of cholestyramine. So, by polymerizing the starting material, the conjugation of starting material, by polymerizing it, we can get the structure of cholestyramine. So, if we see the SAR of cholestyramine, what it will be? Cholestyramine is a copolymer that is consisting primarily of polystyrene with a small amount of divinyl benzene as a cross linking polymer. As I have already mentioned that the cholestyramine is a polymer structure, polymeric structure in which the styrene and 1,4-divinyl benzene is conjugated and polymerized to give us cholestyramine. So, it is a copolymer, primarily polystyrene with a small amount of divinyl benzene as cross linking polymer. It contains approximately 4 milli equivalent of fixed quaternary ammonium groups as of dry resin. The positively charged groups functions as binding sites for anions, increasing the amount of divinyl benzene, which ultimately increasing the cross linking and reduces the priority of the resin, prevents binding of the bile acids to the interior and decreases the efficacy of the compound. So, if there is increase in the cross linking, if there is too much of cross linking because of the 1,4 divinyl benzene, then there will be the decrease in the efficacy of the compound. So, the reaction between styrene, polystyrene and 1,4 divinyl benzene should be in that manner, should be in that quantity that it should contains approximately 4 milli equivalent of fixed quaternary ammonium groups to give it a proper structure which has sufficient amount of efficacy of the compound. And the cholestyramine, it has a high adsorption capacity that cholesterol for bile than cholesterol for bile salt. That means, if cholestyramine is given to the patient alone or in conjugation with the other drugs, it is a very potent compound which treats the problem, that is hyperlipidemic patients, to treat hyperlipidemic patients in a positive way. But what thing we need to get into mind is the polymerization. If there is too much of cross linking polymers, too much of cross linking polymerization because of the group 1,4 divinyl benzene, then it will not that much effective compound. This is the cholesterol, the second compound of this class, that is the synthesis. If we add diethylene triamine with epichlorohydrin, then there is a single step reaction, it is a single step synthesis which gives a cholesterol. As you see the structure of cholesterol, it has too many ethyl groups in it, and it is the simple structure which is made from these two starting material, that is diethylene triamine and epichlorohydrin. These two get involved to make cholesterol. It is a single step reaction. What will be the SAR? The SAR is again, cholesterol is a copolymer. We have seen the two drugs from this class, that is cholestyramine and cholesterol. Both are copolymers of tetra. This is cholesterol, we are discussing about cholesterol. Cholesterol is a copolymer of tetraethylene pentamine and epichlorohydrin, and commercially they are marked as hydrochloride salt. That means there is a presence of hydrochloride salt with it. It makes it more relevant for absorption. So, cholesterol is a copolymer of tetraethylene pentamine and epichlorohydrin, and commercially it is marked as hydrochloride salt. The key functional group of cholesterol are the basic, secondary and tertiary amines. That means the tertiary and secondary and tertiary amines plays an important role in the efficacy, in the potency of the drugs. The total nitrogen content of cholesterol, that is greater than cholestyramine. So, cholestyramine has less content that in form of nitrogen. So, cholesterol is having more nitrogen content in it, which gives it a good, a potent agent, anti-hyperlipidemic agent in comparison to cholestyramine. And quaternization of cholesterol with methyl iodide increases the capacity in vitro for glycocolate. And the SAR usually says that the chemical modification, the structural modification in both the compounds, that is cholestyramine is cholesterol, is in the ring system, in cholestyramine not in the ring system, in the total structure which has the nitrogen in it, the nitrogen content in it, in cholestyramine at the end cholesterol. Cholestyramine and cholesterol both were copolymers. In cholestyramine, there was two starting materials which are in conjugation gives the structure, that is polystyrene and 1,4 divinyl benzene, which gives this structure of cholestyramine. They are the copolymers, and same as in cholesterol, that is diethyltriamine and epoxy chlorhydrate will give this structure of cholesterol. So, there is an huge and very important role of the starting materials. Because of that, there is a polymerization happens in the structure for both the anti-hyperlipidemic agents, they are secutrins. So, what are the therapeutic uses? The first very important use, it will be it can be used in lowering the plasma cholesterol levels in all type of hyperlipidemias. We have already studied the types of hyperlipidemias in our previous lecture. So, the therapeutic use for both the medicines, for both the drugs, that is they lower the plasma cholesterol level in all types of hyperlipidemias. In atherosclerosis, they are also helpful because in atherosclerosis what happens, there is a deposition, there is a accumulation of bad cholesterol in the nerves, in the arteries. So, the blockage causes pressure increase, blood pressure increase, and it can happen, it can lead to several other problems in the vital organs, basically in the heart. So, in patient with myocardial infarction, angina pectoris, stroke, transient ischemia, attacks to lower cholesterol levels. So, by decreasing the cholesterol level, the medicines, the drugs are used in the patients, in these various patients. So, it is very important, the zest of studying the anti-hyperlipidemic agent is to decrease the accumulation of bad cholesterol and increasing the good cholesterol, these diseases are helpful in doing that. And this is the chart which is showing the types of primary hyperlipoproteinemias. So, this has the chart which is showing there are various types of diseases like familial lipoprotein lipase deficiency, the familial hypercholesterolemia, then polygenic hypercholesterolemia, then familial dysbeta-lipoproteinemia and hypertriglyceridemia, familial combined hyperlipidemia. These are the name of the disorders which are they are the types of disorder which are very bad, which are showing very bad effect. Because of that, patients collapsed, are having different types of diseases, lipidemic diseases. So, this is all about in this hyperlipidemia, anti-hyperlipidemic drugs. These are the references for the topic, the principles, the SAR which are taken from Foes. The synthesis generally is taken from the book Rudin, Vardinen and Victor Rudy, and the mechanism of action portion or therapeutic use is taken from the book Essential of Medicinal Chemistry KDT, and some of the parts is also taken from Lippincotts. Thank you very much.